Hello, good afternoon. So what we're gonna do today is to wrap up lecture 5. This is the last lecture of the first half of the course. And let's just start with going through the part points here. We already took the notes on this, but let's just go through the slides and recap some of the key things that we've learned last time. And if you have any questions, please do ask. So the whole process of cellular respiration, as I told you, is divided into 4 separate stages, right? We have the glycolysis, the pyruvate oxidation, as well as the Krebs cycle. So these first 3 steps is where we are going to be oxidizing the glucose. And what that means is we are trying to remove electrons from the glucose, and these are high energy electrons that we can capture using either NAD, NAD plus, or FAD, right? So we capture them and we're going to transport them to the electron transport chain, stage number 4, right? The ETC. And that is where you will be able to harness the energy from these electrons to produce a lot of ATP. In fact, a majority of the ATPs are produced using this method. Right? Hopefully 90% of it is going to be produced using this method, which we call oxidative phosphorylation, right? As opposed to the other method, which is not very common. The other method is called the substrate level phosphorylation. Okay. This is basically what I said, but let's read it through again. All the NADH and FADH2 generated in the first 3 steps. Okay, glycolysis is stage 1, pyruvate oxidation is stage 2, and Krebs cycle is stage 3. All of them will take the high energy electrons to the cristae of the mitochondria, where the ETC is located. As you know by now, ETC is just a group of membrane protein, right? In the cristae. Okay. In the drawing that we have in the notes, I drew 3 of them for you, right? But you can see, like in this picture, there are actually 5. Right? 1, 2, 3, 4, 5. But I just keep it simple, okay? To draw 3. It doesn't really matter. The key thing to know is it allows the electron to be passed down these proteins, and in doing so, you can extract the energy from these electrons. So series of proteins located in the cristae, I just wrote that. And it plays a role in passing down the high energy electrons and harnessing their energy. Okay, so this is the mitochondria here. The blue stuff, it's the matrix. And basically you will have your ETC like all here, right? And you can see why having more surface area allow us to pack in more ETCs, right? So it's like scattered throughout the inner membrane. And here, it's a diagram that shows you how it works. You can think of the electron transport chain as staircase. Right? So the electron, it's like a ball that you drop at the top of the staircase and it just bounces down by itself, right? At the end, you have to have oxygen acting as the final electron acceptor. Right? It pulls the electron away from the ETC. and it allows the next one to fall down the chain. But it begins with the NADH dropping off the electron. Right? The NAD plus picks it up from the first 3 stages, and then the NADH will drop it off. Now, what's missing in this picture is FADH2. FADH2 will also drop off the electron, but it drops it off at like at a lower point, right? So that's why it's not going to be able to generate as many ATPs. All these information are in the notes we took last time. I'm just refreshing your memory on it, right? So this is the picture we drew. We have the, in this case, this is the intermembrane space. And this is the matrix. And it says inner mitochondrial membrane, but we know it forms the cristae, right? The foldings. So NADH is going to drop off the electron, which is going to be passed down. Okay, never mind the skeleton here for now. We'll talk about later. It's going to get passed down. And as it's being transported down, we pump the H plus, right? From the matrix to the intermembrane space. And that creates a high H plus concentration here and a low H plus concentration. The H plus wants to come back in, but the inner membrane will block the H plus because it's a phospholipid bilayer, right? This inner membrane is phospholipid bilayer. So the H plus cannot go through, it's blocked. The only way for them to come back in is through this special doorknob protein called the ATP synthase, or ATPase for short. And as the H plus comes back in, it will turn the protein, it will turn the ATP synthase, and you're going to be able to generate ATP. Okay? And again, what's missing in this picture is that you would have like another energy carrier here. Right? This is FADH2, right? And it's going to have its own electron. And what happens is it will drop them off at the second protein. And then it will become FAD again. Right? So the electron is going to get passed down, but not for as much compared to NADH, right? And so it's only able to pump enough H plus to produce 2 ATP. Okay. Any questions? All right. This is basically the description of what I just told you. You can read it on your own. Excuse me. As H plus pass through ATP synthase, the energy is used to add P to ADP, generating a large amount of ATP. Okay. Never mind this word here, chemia osmosis. Yeah, it's just talking about the H plus moving it down. What's important is this last point. ATP produced in ETC using oxidative phosphorylation, right? Because the energy is harnessed by oxidizing NADH and FADH2. All right, let's do some poll questions. Okay, I only have 2 answers. Has been a minute. Okay, that's fine. Oxygen is the final electron acceptor because it can be oxidized into water. No, that's not true. It's picking up the electron, so it's reduced into water. Oxygen is the final electron acceptor because it's more electronegative than the proteins in ETC. That's true. And more electronegative means it has higher attraction for the electron. Okay, so it has to be stronger attraction than the third protein, right? Otherwise, it's not going to be able to pull it off. The other ones are not true. Okay, here's another one. Substrate level phosphorylation. Where does it happen? Now, I'm not getting a lot of response for these polls. Is it because you guys don't know the answers, or is it because you don't see the poll? I mean, if they're not useful, then I'm not gonna take too much time on them. Okay, the whole point is for you guys to show me whether you understand or not, right? Okay, so I got half the class to give me the answers. And the correct answers are glycolysis and Krebs cycles. Okay, we haven't even learned this last one yet. So glycolysis and Krebs cycle. In the study guide, this one that we fill in. Okay. Both ATPs here and ATPs here, those are produced by substrate level phosphorylation, right? That's the only time. 4 out of the 36. That's why it's 11%, right? Only a small percent of ATPs are produced this way. Okay. Now we're gonna take a look at, you know what? Actually, before we do this, I want to fill in this table with you. This is an important table to go through together. So why don't you pull that out?
Is this an answer like a study guide?
Yeah, it's the study guide, which I will post the answer, right? On eCentennial. I'll do that right now while you guys try to find that. Okay, it's already up, but we can take this out. Okay, so I can do some explanation along the way as well. All right. So in glycolysis, which is the first stage, how many ATPs are produced through substrate level phosphorylation? 2 ATP. Right? You invest the 2 ATPs, and then you produce 4 ATP. So you produce a net of 2 ATPs in glycolysis. Okay, what kind of energy carrier do we produce? And how many? Does anybody remember?
2 NADH.
That's right. We're going to produce 2 NADH. But these NADH is going to have a bit of a problem. Okay, so I'm going to put a star here and I'm going to explain. NADH produced in the cytoplasm cannot get to the ETC directly. Okay. And I can draw a quick picture here for you. Okay, this is the outer membrane. This is the cristae. This is the matrix. And then we have the ETC here, right? And in the cytoplasm, that's where glycolysis happens. Okay, so we have these NADH, which must go here, right, to the ETC, but it can't because like it's too big. It's not going to be able to cross the membrane. Two membranes, in fact, right? I'll let you copy down the picture first, maybe. So it can't go through, cannot go through, and instead, it's just gonna let the electron go through. Okay? And on the other side, we're gonna have an FAD that picks them up. So you might think, hey, why don't we use another NAD to pick it up on the other side? Because no energy transaction is 100%. Every time you move something, you need to spend a bit of energy. Okay, so by the time the electron goes through, they would have lost some energy already. Right? You would have to pick them up using something that carries less energy. In this case, FAD. Right? So over here, even though NADH supposed to produce 3 ATP, but because of the problem I just described to you, they actually get converted into FAD, H2. Right? Not converted, but then like the energy is transferred, right, to FADH2. And so when this goes to the ETC, we multiply by 2, so only 4 ATP. And at the ETC, it's through oxidative phosphorylation, right? Does anyone have any question about this transfer of energy? Okay, so how many ATPs in total? Well, 2 from substrate level, 4 from oxidative. 4 plus 2 is 6. Okay. And what number are we trying to get in the end? What's the magic number? 36, that's right. So this is only 6 of them. So let's keep going. Pyruvate oxidation. How many ATPs do we get from substrate level phosphorylation in pyruvate oxidation?
Zero.
0, that's right. None. If you recall, for pyruvate oxidation, there are only 3 things that happens. Number 1, removing carbon dioxide. Number 2, produce NADH. And number 3, add the coenzyme A, right? So no ATP is produced directly. That's why it's 0. But you do have 2 NADH for every glucose, right? Per glucose. Now these NADH are already in the matrix. That's why it doesn't have this problem. It's already in the matrix. It can just go directly to the ETC. And so we're gonna multiply by 3. That gives you 6 ATP. So just 6 right here. Nothing from substrate level phosphorylation. And then just 6 from the NADH.
Vincent, so the 6 here is because NADH produces 3 ATPs, right?
Each, that's right.
Okay.
Yeah. Okay. For the Krebs cycle, how many ATPs do we make directly? Look at that sheet that I created with you last time. Is it this one? Right. Look at here, the glucose, pyruvate, and then it goes through the Krebs cycle here, right? Right. So look at these numbers, and we're basically putting it back here. Right. So this is 2 ATP. Each of the pyruvate is gonna get, okay, so glucose and then pyruvate, 2 of them. Each of the pyruvate will become acetyl-CoA, and then they will go through their own cycles. Each cycle produce 1 ATP. That's why there are 2 ATPs for every glucose. 6 NADH, look at the summary sheet. How many FADH2?
2.
2 NADH2. FADH2, I'm sorry. Okay, so we're gonna multiply this by 3 and 2. That gives you 18, 4, right? 2 plus 18 plus 4, that's going to be 24. And if you add these up together, 6, 6, 24 is 36. Okay? So for every glucose that we metabolize, for every glucose that we break down, we can get a total of 36 ATP from it.
Excuse me, Professor.
Yeah.
You have said 18 ATP and 4 ATP, so both of them 22 ATP. Why you
Sorry, one more
say
time.
24?
Which
Eight-
one?
18, the Krebs cycle.
Krebs cycle? Yeah, go ahead.
18 ATP plus 4 ATP, so 22 ATP.
That's right. But then there are also 2 here, right?
Oh,
Yeah.
okay, okay.
So together is 24.
Together, okay, thank you.
Mhm. Any other question? Okay. Let's keep going with the PowerPoint. So now we're going to look at a few things that can obstruct, interfere with the oxidative phosphorylation. Okay, that is basically the ETC, right? Interferes with the ETC. So there are things that could block the ETC directly, things like cyanide, right? Carbon monoxide and something called rotenone. There is something that could inhibit the ATP synthase, such as oligomycin. And then there's something that could make the membrane leaky to hydrogen ions, something called DNP. So I'll go through each one of them with you. Let's take a look at rotenone, carbon monoxide, and cyanide. All these things are like poisonous, okay? And they could potentially kill you. They would kill you because they interfere directly with the ETC. So let's take a look at the first one, rotenone. Rotenone is used as an insecticide and pesticide, kills bugs basically. And it binds to the first protein in the ETC. Okay, so I want you to think about the ETC again, the 3 proteins here, right? So if rotenone binds to the first protein and blocks it, then there is no place to drop off the electron, right? So in this case, NADH cannot drop off electron. FADH2 is probably okay because it drops it off later on, like in the second protein or something. But we don't have a lot of FADH2, do we? For every glucose, we only have 2 FADH2. That's not a lot of ATP. All right, so this will significantly interfere with ATP production, which is great if you're, spraying it into insects and pests and whatnot, kills them pretty effectively. But if it makes it into the food chain and humans ingest it, not a good thing, right? Okay, so that's rotenone. Number 2, carbon monoxide. Do you know you're supposed to have a carbon monoxide detector at home? Right? It's like the law. And why is it important to have a monoxide detector? What's so special about carbon monoxide that we need a detector for it? Does anybody know?
Yeah, like when we burn food, so it creates carbon monoxide. Yeah.
Not necessarily, but you're on the right track, I suppose. Anyone else, maybe? What does carbon monoxide smell like? Anybody know that? Do you ever smell
Like
carbon monoxide?
smoke.
Smells like smoke, okay. Anyone else?
I think there is no smell of carbon monoxide.
That's right. Carbon monoxide is odorless. You can't smell it. It's tasteless and is colorless. You cannot see it. Okay, using our 5 senses, we have no way of detecting carbon monoxide. It could be in the room, we wouldn't even know about it. That's why we need a carbon monoxide detector. Okay, it tells us carbon monoxide is in the room. Okay, and usually you get carbon monoxide when you try to do combustion, like burn things, without adequate oxygen supply. Okay, that's why if there is a blackout or something in the winter, people say do not bring in a propane burner and use it indoor. That's going to generate a lot of carbon monoxide. And when you breathe in carbon monoxide, you could die, right? The reason why it kills you is because it binds to hemoglobin. You might remember hemoglobin. We talk about it in the protein structure, but it's in your blood cells, and it helps you transport oxygen. The problem with hemoglobin is that when given a choice between carbon monoxide and oxygen, hemoglobin will always choose to bind to carbon monoxide. Okay, and instead of delivering oxygen, it's just stuck to carbon monoxide. And we all know without oxygen, without the final electron acceptor, there is nothing that would pull the electron off the ETC, right? And then the electrons are going to get stuck. The flow of electron has been disrupted. You are not generating the H plus concentration difference. You're not making ATP. That's why carbon monoxide poisoning is deadly. Here's the thing though. You don't die right away, okay? If you breathe in a little bit of carbon monoxide, what happens is like you will start to feel a little bit tired because you're not getting enough oxygen in the body, right? You might have a little bit of headache. And most people, if you're home alone and you have a bit of a headache, and you feel tired, you might take a Tylenol or something and then go to bed, right? And that is why it's dangerous. If you don't have a detector at home and then you go to bed and there's carbon monoxide, that's how you die, right? So that's why you need to have the detector preferably close to where you sleep. So if you live in a house, a source of carbon monoxide might be your furnace. Okay? Normal furnace, if it's functioning, it's fine. It doesn't have carbon monoxide. But when it starts to malfunction, it's possible to have carbon monoxide seeping out. Also, the car. People say don't sleep in the car while it's running. Like, don't park the car in the garage, leave it on, with the AC on or something, and then sleep in it, right? The carbon monoxide is gonna, from the exhaust, is gonna come into the car and, could kill you as well. All right, last one is cyanide. Cyanide binds to a protein in ETC and it inhibits it. The biggest problem with cyanide, which is what this skeleton head is showing. This is cyanide, CN. CN is more electronegative than O2. Okay. It likes electron even more than oxygen. So with cyanide stuck to one of these proteins in the ETC, even when the oxygen comes, it's not able to remove the electron from the ETC, right? Because it's glued to the cyanide, which in turn is stuck to the protein. So then the ETC gets clogged up again. You're not producing ATP. We die. Okay, so these things are very poisonous. The only one that poses a real threat to everyday life is the carbon monoxide. That's why we have to have a detector. You're not gonna find cyanide lying around, okay? Like, it's like we're not gonna come in contact with cyanide in our daily lives. Are we good? Okay, DMP. DMP was accidentally discovered in a factory, and it was used as like a photographic chemical, like in films, right? Okay, and so they sometimes use it to make a fertilizer, as well as to make dyes and sometimes even explosive. And what happens was in 1934, what happens was there was a factory worker who accidentally consumed a little bit of this chemical. Okay, and he experienced unexpected weight loss, like super quick weight loss, right? And after people find out what happens, the pharmaceutical company thought they hit the jackpot when it comes to weight loss pill. They're like, oh, this is it. This DMP, this substance, right, causes rapid weight loss. We can market it. Okay, and it did enter the market in the 1930s. But they didn't quite understand why it was causing the weight loss initially. It turns out the DMP causes rapid weight loss because it help move H+ across the inner mitochondrial membrane. So again, back to our favorite picture of the lecture. The whole process of ATP production relies on the fact that you can pump H+, right, into the intermembrane space. And you need the H+ to go through the ATPase to make ATP. But if you have DMP, right, what DMP does is it provides an alternate path. It's like, hey, you don't have to go through the ATPase, right? The DMP is going to help you move the H+ too. So that's going to be a problem. That's going to reduce the amount of ATP that is being produced, isn't it? Okay, because they're leaking back in as opposed to going through the ATPase. And so the body thinks you are starving, right? Because you're not producing as much ATP, right? So it compensates by increasing metabolism. It burns even more glucose. And when you run out of glucose reserve in the body, the next thing it burns is fat. That's why it leads to the rapid weight loss. Okay. So it turns out it's very poisonous. If you keep on taking it, like if you get accidentally exposed to it, you're probably going to be fine. But if you continuously take it, you could die. Okay, and people did die. So they banned this thing now. But people can still get access to it through the internet in some cases, like illegally, and that's how people still could die from it. You can look it up on Google. Okay, I think there are some teenage girls in the States, 4 or 5 years ago, they got this pill that was supposed to help them lose weight, not knowing that it contains something like DNP in it, and they die. So here is a summary. We have the rotenone here blocking the first protein. So only NADH is affected. FAD is still okay, but it's not going to be enough, right? We have the cyanide and the carbon monoxide blocking the last protein, okay? Because we're not delivering the oxygen, nothing's pulling it off, or in the presence of cyanide, the electron will not get off. Okay? You understand the difference? Right? With carbon monoxide, there's no oxygen to pull the electron off. In the case of cyanide, the oxygen is there, but it's unable to pull it off. Right? It's different, right? DNP makes the membrane leaky. All the H plus is gonna come in. I don't know why it doesn't show, but all these squares means H plus, okay? And then the oligomycin, it binds to the ATP synthase and it inhibits it completely. Okay, so also very deadly, of course. Counting ATP, we did that already, so that's fine. Next slide. The cellular respiration can use other kinds of molecules besides glucose. We can use different types of carbohydrates. Okay, we can use the fructose, and we can use the galactose, for example. We can also burn fat and proteins, but fat and proteins are going to get converted into glucose in some cases. Your liver has the ability to convert fat into glucose when you are starving. But in some cases, you're going to convert it into intermediate molecules in cellular respiration. So what that means is the protein is going to get chopped up and you have 20 different amino acids, right? So some amino acids, maybe, just as an example, is going to get converted into acetyl-CoA. Okay. Some of the amino acid might get converted into pyruvate. So they get converted into these intermediates somewhere along the way. You don't have to start at the beginning. You could start somewhere in the middle. You don't produce as much ATP, but nevertheless, you are producing ATP, right? So this pathway kind of shows you, okay, so like fat, we know is made up of glycerol and fatty acids, and the glycerol gets converted into something that can be fed into the glycolysis, whereas the fatty acids gets converted into acetyl-CoA. Right, there's a whole bunch of math we can do to figure out how many ATPs we can get from a fat molecule, but I'm going to spare you from that. Okay? Turns out longer the fatty acid, the more ATP you can produce. Right? Each gram of fat can produce sometimes 6 or 7 times as many ATPs as the same amount of glucose. Okay, this is a thinking question. I'll let you read it first. And then I will I'll talk to you about it. Okay, I'm not gonna do the poll. Right. So, cyanide, hydrogen cyanide is poisonous. I already told you why. Because it blocks the movement of the electron, right? It prevents the electron from getting off the ATC, essentially. So it's asking you what will happen to the pH in the intermembrane space. So let's think of the normal situation first. You must be sick of this diagram by now. So normally we pump H+ into the intermembrane space, right? But now the ETC is not working because of the cyanide. And so you will not be pumping H+. So you will have less H+. Here is a hint, it says low pH is caused by high H+. So because we have less H+ in the intermembrane space, that means it is going to be higher pH. Okay? Higher pH. Okay, next, we're going to talk about anaerobic respiration. Cellular respiration is aerobic, okay? Except for glycolysis, everything else that happens afterwards is called aerobic. Aerobic respiration. That means needs oxygen. It cannot happen without oxygen. Anaerobic, on the other hand, does not need O2. So it turns out anaerobic respiration is used by simpler organisms like yeast. And of course, we can use yeast in fermentation, right? To make alcohol or to make our bread rice. So fermentation also can happen in human cells, in human muscle. When oxygen demand exceeds oxygen supply. When you need more oxygen than it's being supplied through your breathing, then we switch to fermentation. So this could happen during strenuous exercise. Okay? Imagine running full speed. We cannot sustain that forever, right? 1 minute, 2 minutes maybe. At your absolute full speed, you can't sustain that very long. Right? So towards the end of it, then you don't have enough oxygen going to your legs, muscles. Then the oxygen demand is too great. Then you switch to fermentation. The purpose of fermentation is to provide an alternate pathway to deposit the electron. Okay, provides an alternate destination to drop off electron. Because the ETC is pretty clogged up at this moment. You don't have enough oxygen. There are still oxygen, but just not enough to pull off all the electrons. So that is like having traffic jam right now. So you want to provide an alternate destination to drop off the electrons so that glycolysis can keep on going. Remember, glycolysis doesn't require oxygen. Right? You can keep on producing 2 ATPs. It's not a lot, but it will keep you going a little longer. So the NADH, instead of going to the electron transport chain, in this case, it's going to react with pyruvate and convert it into lactate. Right? So in human, we call this lactic acid fermentation. So the purpose is to allow glycolysis to keep on going. But in order for glycolysis to keep on going, we need to have the NAD+, right? So the buildup of the lactic acid is what contributes to, like, you know, the soreness and the burning sensation after strenuous workout. All right, so here you can see you start with the glucose, 6 carbon. We're gonna do the glycolysis, producing 2 net ATP, and then you're gonna get your 3 carbon pyruvate. Normally, if you have enough oxygen, then you proceed to pyruvate oxidation and the Krebs cycle and the ETC. But because we don't have enough oxygen, the pyruvate will get converted into lactic acid instead. Lactic acid, lactate, they mean the same thing. And in the process, we will use up the 2-NADH, recreating the NAD+. The NAD+ can go back to glycolysis and keep it going. This is not sustainable. Okay? The 2-ATP will only able to keep you going a little longer. And eventually you're gonna have to stop. Eventually that's just not enough oxygen to keep going. And then you have no choice but to stop. I'm talking about like running at full speed as an example, right? Instead of running, you can think about, you know, hanging on to like a chin-up bar. Okay? You can hold on to it for as long as you can, and then it starts to hurt, it starts to burn, right? And, you know, eventually you're gonna have to let go. Eventually you let go and you fall. Right? That critical point where you can no longer hold on anymore, that's when glycolysis is not able to produce enough ATP to keep up with the muscle work that you are using to hold yourself up, right? When you can no longer do that, you have to let go. The muscle doesn't have enough ATP to keep on going. Fermentation in microorganisms. So yeast, which you probably are familiar with if you bake at all, right? If you bake bread, you need yeast, right, to make it rise. So yeast is going to be performing fermentation. They don't have the necessary machinery to do the Krebs cycle and stuff like that. They don't need to anyways, because they're really small, really tiny, unlike us. We need a lot of energy. And in their case, instead of producing lactic acid, they produce carbon dioxide and ethanol. Ethanol is the drinking alcohol. And this is drinking alcohol. Not all kinds of alcohol can be consumed, right? Do you guys know of any other alcohol? You probably have some at home. Besides ethanol, what kind of alcohol might you have under the bathroom sink or medicine cabinet? Do you guys know what I'm talking about? No? That's okay. The alcohol that you use to clean surfaces, right? Like during the pandemic, people were spraying their grocery bags with alcohol, right? Or like they might be, you know, cleaning their phone with alcohol, right? That's isopropyl alcohol, right? So you cannot drink that stuff. It's gonna kill you, right? And there's also like methanol, people who rub on themselves to alleviate like muscle pain. So not every kind of alcohol can be consumed. The ethanol is the drinking alcohol, and that's the one that's produced by the yeast, right? So of course we can use yeast to produce all kinds of stuff. So that's what yeast looks like. This one is actually budding, it's dividing. And it's the same thing. Right? They do glucose, gets broken down into pyruvate, and then the pyruvate gets converted into ethanol and carbon dioxide. If you are focusing on, you know, making wine and alcoholic beverage, then you would extract the ethanol. Otherwise, you know, the carbon dioxide just makes the bread rise, right? Okay. This is just the similar process. You can compare it with lactic acid fermentation. Okay, so there are 3 phases of pyruvate produced by glycolysis. We have the anaerobic version that creates lactic acid. So this first one here, that's in human when we don't have enough energy, when we don't have enough oxygen supply. The aerobic oxidation is the one that we normally perform. That involves the whole thing, all the way to the ETC. involves the pyruvate oxidation, Krebs cycle, as well as the ETC. That's the one that makes 36 ATP for us, which is the one that keeps us alive. The other kind of anaerobic respiration happens in simpler organisms like yeast. Okay, they produce carbon dioxide as well as ethanol. We do not do alcoholic fermentation. Ethanol is actually toxic to our cells. Okay, lactic acid, it's not great, but it's okay. It builds up in the cell, in the muscle, causes some soreness, but it's not toxic. And that's it. That is it for the entire lecture 5. We actually spent like 3 classes on it. If you think about it, it's quite a bit. So no class on Monday, and the test is going to be on Wednesday. Now I have posted a few things for you to do on your own. So you can go to the course homepage, and that is the test information. No class on Wednesday either, so I won't be seeing you next week the whole week. Okay, Monday is family day. Wednesday you do the test. Here's the quiz link. 44 multiple choice questions. 10 multi-select. There will be no short answer question this time, and you will have 100-minute. Okay, 100-minute minutes to do the test, and it covers lecture 1, 2, 3, 4, 5, as well as the Beetroot Lab. There's a practice self quiz that you can do. Let me see. You're only allowed to do it 5 times. Okay, so study first before you do it, because I notice people just keep on doing it, like they're guessing the answers, okay, until they get it right. I don't want you to do that, okay? I want you to actually think about it and try to do it properly. So only 5 attempts allowed, and of course they don't count for grades. Okay.
Vincent, can we watch Beetroot Lab again? Is it possible?
Yeah, sure. But like which part? Like all the explanation and stuff like that, you mean? Or just the
Yeah,
Beetroot?
or information about it, because I didn't write it down.
Okay. Yeah, sure. I'll find it. Okay, and I'll post it for you. Okay. But in terms of the Beetroot lab, hold on. If you go to content, I added a new folder called test review. So you can download that. And these are the only questions you have to know for the test in terms of the Beetroot lab. Okay, so maybe you don't need to rewatch it. Okay, so these are all the questions that you should go through to help you study, right? Okay, but like this is obviously not going to be the only thing that's on the test, on the quiz. You got to do more. The answers are posted as well. The answers are posted like right here. And then I will also post like a review video. Okay. Like this one I did last semester, right? So it's just me taking up the questions with the class. So you can watch that. I was gonna do this on Monday with you, but then like Monday is holiday, right? That's why you can just watch the recording yourself. There is the poll question collection. Okay, like again, I have to say I'm a little bit disappointed with the participation with the polls during class, but that's okay. You do you. This is all the poll questions in one PowerPoint slides. Okay, if you download it, onto your computer, the answer's at the bottom. It's not on the slide. You have to download it to see it, okay? Like the answer for each of them is in the notes section at the bottom. Okay, because as you know, some of these questions show up on the test, right? So you can go through all of them. There's like 40 questions. It's good practice. Do you understand?
Excuse me, Professor.
Yeah.
It's possible that we have access to our quiz too, the question.
No, yeah. So like as I said at the beginning of the course, right? Like we, as in like everybody who's teaching the course, decided that we cannot give the test out because
Okay.
yeah, the questions needs to be protected, right? Yeah.
Okay,
So
thank you.
sorry about that. So that's basically all the resources. Does anybody have any questions? Okay, I'll post another announcement to link to all these things. But there's the self quiz, there is the review sheet, which you definitely should do on your own first before looking at the answers, right? Yeah.
I have like something comment,
Sure.
or maybe I don't know. It's completely from me. It's not
Yeah,
on
it's okay.
behalf of other people.
Sure.
But when I study or read your maybe quiz, self quiz, or the one you post puzzle, all I did is direct. It's easy to understand. But your test, I mean your quiz, is completely different for me from that. For example, self quiz, a puzzle, and other the question, and also the one you post, like resource for reading. I read that too. So it's hard. So why it's like this? It's to for example, I came here to learn and to understand.
Yeah.
Yes. So the exam, I don't know. It's very hard for me.
I wouldn't say the quiz questions are completely different. I know they are not necessarily, identical to
I didn't say
the
that.
self quiz, but
Yeah.
hold on a second. Let me show you some of these things. Okay, let me just
Vincent,